Thank you. Thank you for giving me opportunity. Can you see my slides? Huh?
Yes, we can see it.
Thank you. So I'll be presenting a brief overview of DNA editing techniques and current and future with some examples. So the overview of my talk will be brief introduction and different type of editing technologies. There are multiple editing technologies, but it's beyond the scope of this presentation to cover everything. But I will try to cover couple of main things. And as you know, all the speakers spoke about how these genome editing technologies are used in gene therapies. But I will switch to show you some data where we work on CAR T cells, how we use these genome editing technologies. And a brief overview I will give you, because I think Professor Haider Fragol already mentioned about genome editing technology and hemoglobinopathies. So I'm doing some work with collaborators in India and UK so on the hemoglobinopathies. So I will be just briefly give. We do not have much data on that. We recently started that work, and then I will briefly introduce future of genome editing. So okay. So genome editing is a powerful tool to introduce, our additions, deletions, and substitution in the genome of living organisms. And genome editing involves precise manipulation of cellular DNA sequences to alter cell fate and organism traits, and has the potential to both improve our understanding of human genetics and cure the genetic diseases ultimately. So there are four main families of DNA design and engineer nucleases. Historically, we were using one is meganucleases, and second is zinc finger nucleases, and third is transcriptor activator like effector nucleases, TALENs, and the fourth is a very popular one is a cluster regulatory intercepted short palindromic repeats, which we call CRISPR-Cas. So as you most of you already know that we already aware of genome editing techniques since 1985, but until now recently we started to understand the potential of these genome editing technologies, how we can utilize this for our actual purpose, and to cure the genetic diseases. And most of the genome editing technologies are existing since 20 years back almost. Okay, so this is a snapshot of history of genome editing, and this is another snapshot of just CRISPR-Cas. We know the CRISPR-Cas identified in 1989, as Dr. Froglo either mentioned, but its application only recently started in 2011, where we started to understand how we can edit the genome and precisely and with for our benefits. So it took a long way to go long way to come from identifying and developing these CRISPRs. So I will give brief overviews of these meganucleases and all the other genome editing tools. The meganucleases are also known as the home nucleases. These the long DNA binding recognition sites it has, which are from 14 to 40 nucleotides. Due to its highly specific and long recognition sequence, it is not applicable in modification on a target site in the genome. And due to that reason, it is very difficult to modify or precisely use for specific needs. So that's why they did not get much attention as compared to other, genome editing tools. And it has been shown that they're likely to show off-target binding of genome and off-target effects. And other thing is the lack of readily available customizable reagents and lack of clear rules for designing these meganucleases can hinder the applicability of these meganucleases. The second class is zinc finger nucleases. The zinc finger is an artificial restriction enzyme bound to a fork-1 domain dimer of DNA cleavage, which leads to the DNA double-stranded. All these genome editing tools, leads to double-stranded break or single-stranded break, definitely. So they combine this and engineer together with zinc finger plus nuclease activity of FOC1 to create zinc finger nucleases. The mechanism of target DNA recognition is based on the DNA protein interactions, and the length of recognition DNA target is 9 to 8 base pairs for these zinc finger nucleases. But unfortunately, these zinc finger nucleases are extremely challenging to engineer or design for specific sequences. That's why the popularity is not that available. The popularity is not there for these. So now the third DNA editing and machinery is the talons, a class of DNA binding proteins with predictable specificity. And these talons are also like similar to zinc finger nucleases. They are fused to the FOC1 catalytic domain to create this engineered transcription activator like effector nucleases. Typically, they are 30 to 40 base pair, bases in specificity, and FOC1 cleaves results in double-stranded breaks similar to zinc finger nucleases. However, unlike zinc finger nucleases, these talons recognize DNA triplets. Each tail repeats recognize only one single base pair compared to zinc finger nucleases. The last one is the most popular one is the CRISPR-Cas system. It has two components. Approximately 20 nucleotides, a small guide RNA, which is bind to a target DNA sequence, and a Cas protein, which exhibits nucleus fusion, nucleus function. So the main CRISPR-Cas system which we use mostly is a Cas9 from Staphylococcus pyogenes. It is a ribonuclear protein. This protein is an endonuclease with the two DNA cutting active sites and binds to the guide RNA sequence that are complementary to the target DNA which you are trying to make a double-stranded break. So the most important part is that the mechanism of target DNA recognition is based on the DNA RNA interaction in the nucleus. The length of the recognition of the DNA target is 22 base pair plus the PAM sequence. It's really important to understand this, the genome editing. When it comes to genome editing, most of the genome editing tools makes double-stranded breaks. So in our cell, what happens is once we make this cut with any kind of genome editing tools, there are two immediate repair pathways takes place. One is non-homologous enjoining, which is highly error-prone. The cell will try to repair within minutes or within hours to repair that deletion part, and by doing so it creates a lot of errors like indel mutations, gene deletions, gene insertion or replacement, mass replacement. The second part is the second pathway is the homologous recombinant is a HDR pathway, homologous recombinant pathway. So this is error free. So during this pathway, you can provide a template which is homologous to the template which you are cutting, and you can replace the gene. You can replace the gene, and you can make the gene to be corrected, or gene insertion can be happen also, or gene replacement. However, as I mentioned previously, most of the genome editing technologies are error prone because DNA double-stranded repair is dominant, and non-homologous enjoining machinery functions throughout the cell cycle. But homologous direct repair is linked to replication, so it's only active in late S phase or G2 or M phases. So if the goal is to understand or undertake destructive gene editing, then this will be an easy, for non-homologous enjoining. But however, if the goal of the experiment or goal of the, genome editing technology is to undertake accurate DNA editing, then somehow we have to deal with the unwanted non-homologous end joining mediated mutations when we use these genome editing technologies. So, as I mentioned, previously, when we talk about genome editing, about CRISPR-Cas applications, the most important negativity comes is how we can lower the non-homologous end joining repair, or homologous, how we can increase the efficacy of homologous directed repair, but compared to non-homologous end joining. But HDR occurs at a relatively low frequency and in non-dividing cells. This pathway is further down-regulated, unfortunately. For example, such as, if you, this is, we have extensive experience of using hematopoietic stem cells. If you want to, provide a template after cutting with this genome editing technologies, provide a template to repair this pathway by homologous directed repair. The efficiency of the correcting the genome is very, very low. So what scientists have designed, rather than trying to improve the HDR technology, scientists have developed two classes of other base editors, which is cytosine base editor and adenine base editor. So what they do is, instead of making double stranded break, they will just convert the C to A or A to T. So that's the importance of genome other base editing technologies. So instead of making double stranded break, they will just convert, because it has a cytidine deaminase enzyme called apolipoprotein mRNA editing enzymes, which will convert the the C to A or C to U changes. So there are two kind of advanced base editor, which is, as I mentioned, cytosine base editor and adenine base editor. So cytosine base editors are based on the generates uracil, which base pairs as the thymidine in the DNA. So replacing the CG to TA substitution, and the adenine base editor replacing AT to GC substitution. However, most of the mutations in the human genome, 60% of the mutations in the pathogenic mutations in the human genome can be corrected by base editor, but remaining other transversion mutations cannot be corrected. So the other scientists have also developed the prime editing, which is modified Cas nuclease, which has Cas9 nickase, and fused to reverse transcriptase, and it has a PEG RNA, which is a which is a prime editing guide RNA, which makes the nick in the DNA. It doesn't make double-stranded break, but it makes the nicks, precise nick where you want, and it will change the mutations to desirable, whatever you want to insert or delete from that place. This is one of the popular technology. So this is just overview of the difference between CRISPR-Cas editing with double-stranded break, base editing, and prime editing. So the components of the CRISPR-Cas editing is Cas9, guide RNA, and donor DNA. And base editor, you have fusion of modified Cas9 nickase, and deaminase enzyme is always there to deaminase that, and is the guide RNA all attached to that. But prime editor, fusion of Cas9 nickase enzyme plus reverse transcriptase, then PEG RNA. Similarly, the other differences are you can see these ones. So, as I mentioned, I think most of the talks we have shown nicely other applications of gene replacement therapy or gene editing technologies. But I wanted to talk about how we can utilize this genome editing technology precisely to make CAR T cells, off-the-shelf CAR T cells. So just briefly want to introduce you why we need off-the-shelf CAR T cells compared to bespoke and autologous CAR T cells, because of the cost, and there are multiple reasons like full virology screening not available, and the patient manufacturing can fail multiple times. So and other things are like product viability, quality control. There are a number of I listed whatever we we face in clinical trials, phase 1 clinical trials. From my experience, I have listed all the points here which will be overcome by off-the-shelf CAR T cells. However, for making off-the-shelf CAR T cell, there are some other barriers such as graft versus host disease. So for off-the-shelf CAR T cells, you are taking a healthy donor T cells and trying to engineer them for expression of CAR, for example, CAR19. Okay, but how do you get rid of graft versus host? So you can precisely make a point mutation in these T cells for alloreactive molecules such as TCR, MSC class 1, or for CD52 for alemtuzumab to resist for the alemtuzumab chemotherapy for the patient. So this kind of modification we can make for CAR T cells to get the to make the off-the-shelf CAR T cells. So I will give you best example. First in human trial we use talons to make allogenic CAR T cells in UCL. We have utilized this technology, but it was very laborious because this method we use on the conventional CAR T production like Kymriah and Escata, because those products are made on bioreactor called Zuri bioreactor, not on the Prodigy. So is a very laborious method. So you get the entire procedure takes 20 days. So what we do is we take 1 billion cells on day 0 and transduce them on day 7, activate them, and after 48 hours we transduce them for expression of the CAR. Once they express the CAR, we remove the alloreactive molecules by CRISPR-Cas or TALEN. Here we are using TALEN because at that time we were using this from the company called Selectus. This project was done with the collaboration with Selectus. It's a first human study. So the reagents were proprietary of Selectus. So we use CRISPR-Cas to precisely knock out CD52 for talons to knock out CD52 and TCR alpha beta. So once you remove those molecules, we expand the cells from day 5 to day 17, and then on day 17 you deplete the cells on TCR alpha beta negative fraction. So once you remove the TCR alpha beta positive cells, you collect the cells only CAR positive and TCR negative T cells and CD52 knockout cells also. So those cells are frozen and given to the patients. This is the one best example, first in human allogenic CAR T cells using the genome editing technology combined with CRISPR chimeric antigen receptor expression. So this is just a brief study design on how we treated the patients. Day 7 until day 7 we were lymphodepleting, and then day 0 only a single vial infusion was given to children. And we treated, I think, 5 patients. All 5 patients has achieved complete remission, and this was one of the remarkable results for us that time. And after that, that trial has gone to the selectors completely, and they open multi trial sites. Now the trial is still ongoing. So that was the best example of how we can utilize the genome editing talents for generating off-the-shelf CAR T cells. So this is again, as I mentioned, this was the collaboration with selectors. So, however, using talents, there was not, we were not satisfied with the number of things like the efficiency of TCR knockout is not that great, because the reagents were supplied as electroporation to knock out alloreactive molecules, and the TCR positive carryover was at least 5%, which is kind of enough to cause some kind of a toxicity or GVHD, I can say. But our goal was to see how we can further utilize this genome editing technologies to reduce the TCR positive carryover cells and precisely knock out these TCR and other alloreactive molecules, and how we can also same time automate this technology of producing CAR T cells. So, because as I mentioned, we were using conventional methods to produce these cells, a lot of bioreactors and Clinimax Plus to deplete the cells on TCR and electroporator, um, BTS electroporator, all those machines. But then we have what we wanted to do is we wanted to produce the off-the-shelf CAR T cell, but this time using more advanced method like CRISPR-Cas. And this one we use Prodigy to optimize the laborious procedure on this completely closed system. And on top of that, we combine this electroporator called nuclear factor from Lonza to deliver this guide RNAs and CRISPR-Cas reagents into T cells. So just to explain you how the system works here is that the guide RNAs are we combine the guide RNAs in the vector, which is a third generation lentiviral vector, and we we combine the Cas9 mRNA in the vector plus guide RNAs in the vector, and then we electroporated them and then specifically knock out TCR and CD52 to generate off-the-shelf CAR T cells. As you can see here, on day 12, we had achieved in pure cells, which is approximately 3 billion cells of CAR positive and TCR negative by using CRISPR-Cas genome editing methods. So what we are doing is we are precisely making the deletion in a DNA so that the gene is completely knocked out, and we are not, again it will non-homologous end joining, and the complete genome is complete the expression of the gene is knocked out. So we are not doing anything extra than just sending these CRISPR-Cas reagents to make the cut. I'm not going to go detail, but we have achieved very extremely pure cells, which is less than 1% TCR positive carried compared to 5% using talons at that time, and this one was further reduced later in GMP. So this one further reduced in GMP to 0.2% TCR positive, and this trial is ongoing, and it was very successful, this project, because we managed to get ultra pure allogeneic CAR T cells by knocking out TCR and CD52 so that the alloreactive molecule is gone. And we also managed to use umbilical cord blood T cells to generate these off-the-shelf CAR T cells, which are specific for CD19. So this trial is still ongoing. We treated, I think, 5 patients. So the importance of CRISPR-Cas is good, but how we can get rid of some, I'm not mentioning other things.
2 minutes. You have 2 minutes.
Okay. So the importance of double-stranded break is really here because it can cause off-target effects, a lot of off-target effects. So there are base editings which will not allow double-stranded break, but it will change the single DNA sequence. So what we did is we further gone to utilize these base editors to create a CAR T cells, off-the-shelf CAR T cells, which can target T cell leukemia. So that's the called T versus T. However, there is a huge amount of problem here. We have the T cells also have the CD7 because and they can cause fratricide each other if the CAR is expressed on that. So what we have to do is we have to remove CD7 first along with CD52 and TCR. So then introduce the CAR so to avoid the fratricide, and then generate the CAR T cells which are knocked out for multiple, effectors like CD52, CD7, and by using just, by introducing a single premature stop codon in their sequences precisely. So we able to generate that. We able to generate extremely ultra pure cells for CD7 CAR T cells, and this clinical trial is ongoing. We treated two patients so far, I think, in UCL. So this trial is ongoing. You can see the mice is clearly clearing the mice, for the tumor. So because of the time and as already Dr. Haider mentioned, these genome editing technologies are also can be used for other diseases. So I'm collaborating with a lot of couple of people in India, which is my good friends. Professor Sandipta Bugla has a very good lab in Usmania University, and she has a very good talented postdoc Swati to work on this. And we have a good, Dr. Prashant Hivakar, who was a PhD friend in UCL. We have worked together. And Professor Aris, who worked with me on number of nanoparticles. So we are using all these three technologies. I'm not going to do it. It's a very preliminary, we started this project. We are going to utilize all kind of tools to see how we can use the genome editing approaches for sickle cell disease. Inshallah, by next year, I may have some results with this. We have recently started this, preparing the reagents to clone and start our preliminary work. And most importantly, we do have access to patients, mainly beta thalassemia patients and sickle cell disease patients, through Dr. Prashant. And there are a lot of patients, and need unmet need for these things. And Dr. Aris has a proprietary, nanoparticles which can be delivered these, editing tools. So I just want to skip through what are the other future toolboxes, in emerging tools, the CRISPR-I, the gene repression and gene activation, CRISPR-R, CRISPR-A, and other technologies that are emerging, CRISPR-Cas3 and associated transposase and Evolor. Okay, so by that, I will like to thanks for listening to this talk. Thank you so much for listening. Thank you. I'm ready to take any questions.
Thank you very much.